Here we go. DNA replication in 3, 2, 1.
DNA replication is the process of producing two identical replicas from one original DNA molecule. This biological process occurs in all living organisms and is the basis for biological inheritance.
In a semi-conservative process, in a process that produces two copies of strand of DNA that contains an old and new strand, DNA is split by helicase in an anti-parallel organization, but this is after DNA is unwounded at the replication fork. This is also where the original replication is, where the replication sequence is first initiated. But before all these primers, needs to synthesize primers to serve as a starting point for DNA synthesis. The leading strand is the 5' end, whereas the lagging strand is the 3' end. Everything is perfect on the leading strand because DNA polymerase is able to add new nucleotides to the leading strand. And because the new nucleotides can be on the 5' end.
The primary enzyme involved in DNA replication, DNA polymerase III, extends a short RNA primer which is synthesized by RNA polymerase onto the lagging strand.
The discontinuous lagging strand replication with elective fragments are linked together by RNA primer, which is just a bit of RNA, just so you know. Love.
Once primer is down, DNA polymerase can run as opposed to like it does on the leading strand, but DNA polymerase cannot bind with the 5' end right, so DNA ligase facilitates the joining of DNA strand together to clean up the gaps. Done!